as Giovanni already mentioned, so we actually had started a project shortly before everything was closed down. So we had a little bit of time already to prepare. And then, of course, the big challenge was to keep things running while chemicals would not be delivered anymore to the university. And so there was a core team of 3 people of my lab who were involved in this and who were still able to do some work during the shutdown. And I will here present what the outcome of this work is, and hope that you find it interesting. So first of all, I gave a talk for the Zukunftskolleg that was already 2 months ago. And at that time, we still had around about 3.5 million infections of coronavirus worldwide, and the deaths was approaching 250,000. That was the 5th of May. Now the numbers have changed dramatically, so we are now at 14 million infections roughly, and approaching quite soon 600,000 deaths. To put this into a comparison, you see here the death rates of other important reasons for deaths. For example, malaria is close to what we have now for coronavirus, but for the whole year. So we are now at a point where we have within 4 months around about 600,000 deaths worldwide, and it's still, as the graphs show, it seems not to back down, but rather continue like this. So with this, it shows you it's one of the major health-related problems currently in the world, and of course not only by the death rate, but of course also people who had been infected, and this will probably turn out within the next couple of years, only when more research on this is available, what kind of delayed effects of the pandemic and of coronavirus infections will there be on individuals that have been infected. There are already now known that there are some neurological problems that might turn up. There might be some reduction of lung volume, of general health conditions that might be very problematic even for those who survive the infections. Also, of course, we all are aware of these major economic problems and social problems that the pandemic brings about. And so, of course, we are all striving to find somehow solutions, or to at least come to a better understanding of the virus. Speaking of the virus, we always call it the coronavirus. In fact, it's one member of a large family of coronaviruses. So there are many of this genus Coronaviridae, and it's in the subcategory of beta coronavirus. And you see there are even coronaviruses that commonly affect us. For example, 10 to 15% of the common cold is a coronavirus, a very different coronavirus though. And there are also different coronaviruses. You have heard probably of MERS or SARS, which already have been at least epidemic in a much more restricted way. And you also can see from this that a lot of coronavirus species are in bats or in certain animals. So it's spread over from cows and horses to birds and bats. So there are a lot of different species that can get coronaviruses and then are a reservoir for coronavirus infections. But what we are now focusing on is this one coronavirus that's important for the current COVID-19 pandemic. So what does coronavirus do? If it invades the human body, so it first is this coronavirus particle. It's this virus particle, which is like a lipid membrane shell, and inside packed the genetic information of the coronavirus, which is RNA, and then it hits a surface of a human membrane. So this is a human cell, and here are some receptors, and it attaches to these receptors, and then there are different possibilities how it can melt together with the membrane or being endocytosed and then opening up. And then so it basically gives free its genomic information into the human cell. And this genetic information, the genome of the coronavirus, then starts actually doing its pre-programmed job. So it's not an actual living being, such a virus, it's debatable what it is. So it is basically just a genetic information that's engulfed in a lipid protein vesicle, and that contains tightly packed genetic information. So that's the genome of coronavirus if we look at it from the functional perspective. So there are different open reading frames. That's always what is being directly translated into a protein. And you can see that there are different genes that are encoding for different parts of the coronavirus. If we look more closely at it, then we see that there are actually only 4 of these genes that encode for the proteins that make up the coronavirus particle. So there is this spike protein, which are these prominent spikes on the surface of the virus, which gave it also the name coronavirus, because in electron microscopy you see then like a corona-like structure or some glare around it. Then there is the M and the E protein, which are important membrane proteins. And then there is this N protein, which basically wraps up the whole genetic information inside of this vesicle and packs it. So it's basically just like a bag for the DNA, so that it's tightly packed and well preserved. So all of the rest of these genes that are in the genome of the coronavirus encode for proteins which are non-structural proteins, and they are important for the replication of the virus. So actually everything that's happening is happening in the eukaryotic cell, so in the human cell. It needs the host cell in order to turn into an active state. Before it's just basically a passive particle that cannot do anything on its own. And so all of these proteins that are here encoded, they are important for the replication of the virus in the cell, for hijacking the infrastructure of our own cells to make new virus particles. And there are 2 important things that actually caught our eye in the beginning, and that was right before the pandemia broke out. That was these 2 proteases, which are encoded in the genome of the virus. So there's the PL-Pro and the 3CL-Pro. That's 2 proteases. I'll come to that shortly to explain what a protease does and why it's important for the virus. Now, the activity of a protease is in principle being like a scissor that cuts off a protein at a predefined sequence. So this is like a peptide bond of a protein, and then comes here the enzyme, the protease, and what it does, it hydrolyzes or just cuts like a scissor here between these in this bond, and you get like half of a protein or a chopped off protein and then the rest of it. Okay, so why does the virus need these proteases? And that's actually the trick of this virus, that it produces most of its proteins that are required for its replication as one large polyprotein. So it's a super large strand of protein, which is yet not mature, and the individual components of it cannot work. And they cannot work until they are chopped off by these proteases. So it has its own proteases that actually cut off the individual parts of the polyprotein so that these individual proteins become active and can work and do their job in the cell to replicate the virus particle. And for this, these two proteases are very important. Additionally, the PL-Pro has some activity in basically escaping the human intracellular defense mechanisms by avoiding that the proteins that are produced of the virus are targeted to degradation by the human host cell. So it actually defends the virus from being destroyed inside the cell, and the other protease mainly does the job of activating all of these previously inactive subcomponents of this super large protein by chopping them off. So this is how this one protease looks like, the 3CL protease. And you can spot in there, there's some kind of special structure, and that's what we call an active site. So a protease consists of like a cleft, an active site cleft or pocket, in which then this sequence that it needs to cleave can bind. And it additionally has a certain structure of amino acids which allow to do this catalytic mechanisms of cleaving of a bond. And that's what you can see here. So that's actually the only chemistry slide that I will show here. And you have here the peptide bond, and it gets into this active site pocket. And there are these two amino acids which catalyze the reaction of cleaving off this part of the protein and then liberating the other part of the protein. And you just need one water molecule for this reaction to complete, to have basically cut apart. And that's happening of positions here. So here will be cut, so the protease actually cuts out, and then it cuts all of these other which are important. What we thought when we noticed that there is a cysteine or two cysteine proteases in the genome of the coronavirus, then we immediately thought we could use that with our currently being developed chemical probes to see if we can find small molecules that actually can bind to this active site and label it. I will show you later on what actually labeling of an active site has benefits or advantages or applications that they could be useful for. You can use this, for example, to use a fluorescence marker, which is basically something that then lights up under fluorescent light, and then you can tag it by something that's called click chemistry, which is actually just like playing Legos. So you have one Lego building block and another, and you click it together, and then you have a little bit larger block. And if one of this is a red Lego block, then you can see it, for example, easily than if it was just a gray one. And that's what we basically do. We take a fluorescent marker which can emit fluorescence as a signal and thereby label it so that we can actually see what's happening there and if we can actually target this protease. Now, so how do we get actually a protease of the coronavirus? The good thing is that nowadays genomes are worldwide openly available in the internet. So they are published and accessible for everyone for free. So you can just search for the genome of the coronavirus, and then you get here all of the genetic sequences of the coronavirus, so all of the nucleobase pairs that make up actually the whole genome of the virus. So this is just a small fraction. So this would be many, many pages of this ATCG and so on. Then we selected the sequences that we were interested in, so the sequences that encode for these proteases. And then we did some codon optimization and added a purification tag, and we also made an active site mutant. And then luckily there are nowadays companies which actually can synthesize any gene that you want for quite cheap. So you can get for like 10 cents per base per a gene being synthetically made. So this is the kind of new world of synthetic biology where you can synthesize any gene on demand that you want. And then we got it cloned into a plasmid. And so we have this and could transform it in E. coli cell. So that's Escherichia coli. It's a intestinal bacterium, which is actually the workhorse of modern biochemistry and molecular biology labs. And you can put such a construct, such a plasmid that has this gene of the virus inside of E. coli and force E. coli to make a virus protein that actually E. coli would normally not make. So that's what we did. And then you run, and you will see now a couple of these gels. That's why I will shortly explain it. So if you have all the proteins that an organism produces, and you want to see a certain special protein, then you run gel electrophoresis, which means you separate actually all of these proteins according to their size. The largest ones are at the top, and the smallest ones are at the bottom. So you have here this protein size distribution, and the intensity says something about how much of this protein is there. So this small band corresponds to a low abundance protein, and this thick band to a probably higher abundance protein. So now we did this for the coronavirus proteases, which we expressed in the E. coli cells. And you can see here these thick bands. That's a special colorant that has been used to make them visible actually to the eye. And you can see here a thick band for the 3CL-Pro, the one protease the coronavirus produces, and here another band for the other protease that the coronavirus produces. So this is just basically protein abundance, and the rest here is all E. coli proteins. So every small, even the tiniest bands here that you barely can see are some proteins that a normal E. coli cell has. So now we wanted to make chemical probes that actually can selectively label these proteases and only label them at the active site, which is important for cleaving a peptide bond. So what we did is we were searching for chemical probes that can do this job and only bind to this coronavirus proteases in a specific manner. And that's what we did. So we searched for these probes. And first of all, we found it's extremely hard to find probes that are specific for it. And so with this fluorescent labeling, you can then see like a fluorescent band if it has bound to the protein. Whereas when you stain it with this general protein stain, then you see basically all proteins, regardless if they are fluorescence labeled or if they are just not labeled. And so in order to find probes which can actually do this job, we employed a new strategy which we developed. And this is what we called a ligand selection probe. So we have here these ligands, which are some chemical structures, and they can now be combined with this probe scaffold to give a great diversity of different probes. So with this, we can, within very short time and very efficiently, produce diversity of chemical probes, because some probes might not fit into the active site and others might bind somewhere else, and some might not bind at all. And so with this strategy, we actually could more rapidly find probes that actually are active. So what we did is we had our E. coli cell here, which produced this coronavirus protein. And then we added our specially modified probe with one of these modifications here. And if this probe could bind, then it was fluorescently labeled by this click chemistry. So you remember the Legos, so you put them together, and then you have here this fluorescent signal tag stuck to the probe, and the probe is stuck to the enzyme that we're interested in. And if everything would go well, we would see like a band, a fluorescent band only for this protease. And if we had a mutant in which the active site is basically destroyed, then we should see no labeling. If we still saw labeling here, then it would mean that this probe is unspecific and binds to some site which is not the active site that's required for cleavage for cleaving a protein. So it's actually then not a specific probe, an unspecific probe. If we see no band at all, then it would mean we failed and we could not produce an active probe that can label this protease. So here you see our actual results with so every LS with a number here corresponds to a chemical molecule, which is one of these probes. I will not show the structures now here, but you can see some of them were unspecific, so they did not specifically label the active site, but some of them were quite specific and only labeled the active site that was intact, but not the mutated active site, where we actually just replaced a few amino acids so that it would not be an active protease anymore. And we also could do this for PL-Pro, so the second protease of the coronavirus, and also find here a few examples where we saw a high intensity of labeling by the probe of the wild type and a very low intensity of labeling for the mutant. So this shows you that there are specific probes that only address the active site. Here it's summed up actually for all of our probes and combinatorial library that we made. And you can see that some of them here showed basically a good wild type to mutant ratio, whereas others were completely unspecific and only labeled, for example, the wild type, but also the mutant equally well, so that the wild type mutant ratio would then be extremely low. And so we picked out the best ones that actually could specifically label the active site of these proteases. And here you can see a whole gel in the background of a native E. coli cell. And you can see that actually the probes not only were quite specific for the active site of the proteases, but also selective for the coronavirus proteases, because there are many other proteins that you cannot see here, but that you can see in this general stain that actually stains all proteins that are there. So here you can see only this protein of the coronavirus in the whole background of the native E. coli proteome. So it's a very specific and selective labeling that we could achieve with these probes. Now we were wondering what could be applications of these probes. First of all, we were interested because E. coli is of course not the ideal system to study, because normally E. coli cells are not infected by the coronavirus. It was just for us a model system to make easily these coronavirus proteases. So we actually purified the coronavirus proteases and added them to the lysates of human cells. And here you can see it. There are still a lot of other bands that you can see in this case, but you can still identify here the coronavirus proteases in the background of a human cell, which is very important because now you can use these probes as chemical tools in order to investigate in more detail the activity and the function of these proteases even inside a human cell or in the background of a human proteome. The second thing that we were interested in is because we noticed that the protease, especially the 3CL protease, is first in a non-active state and then starts to cleave itself off from this long polyprotein, and thereby it activates itself. So it means here a part of its active site is actually blocked by this rest of the protein. So this is a simulation which our collaborators did, and that show that it can occupy different possible spaces here, and but it always blocks a part of the active site. And that means that with the typical way of finding new drugs that could block the coronavirus protease, you could not study this pre-activated stage of the protease. So we were lucky that actually our probes were able to label the active site despite of this. And that means that we have now a perfect tool at hand with which we can find new inhibitors in order to target the coronavirus protease and possibly and use them for searching for new drugs against coronavirus. So what we did is in order to see if actually this was true and if we could use our probe to find potential inhibitors of the coronavirus proteases, we incubated the protease with potential inhibitors. And then if an inhibitor would bind to the active site and block basically the activity of this proteases, that would then mean if it did this in a live human cell that was infected, that the virus could not replicate anymore in the human cell, and the virus would basically be inactive, and the patient ultimately would be healed. So this could directly result in drug scaffolds. And so we pre-incubated the purified proteins with these potential inhibitors. And if an inhibitor bound now to this protease, then the probe could not anymore bind into the active site. So here you can see it blocks actually the active site, whereas here where you have nothing in the active site and the active site is still fully available, the probe will bind. And then you do this Lego clicking here together and you get a fluorescent tag, but here not, because you still have this bulky thing there that blocks actually access of the probe. And then it would result in the one case in no labeling, and in the other case you would see a probe labeling. And that's what we did by going through thousands of chemical structures that we found and buying always those of them which we thought from a chemical perspective that they looked interesting and potentially could bind to the active site of the proteases, and additionally compounds of which we knew that they already either were approved for human use or compounds that were actually natural products that we could assume would do probably well even for human cells, so that they would not harm the human cell. So that's what we did, and here you can see the percent of inhibition of labeling, so actually how well the labeling by the probe was excluded by one of these compounds. And these are all the compounds, and most of the compounds could do nothing and had no activity in blocking the labeling. But one of these compounds here, and this one in a little bit lesser potency, could block actually the activity or the labeling of the coronavirus protease. We also found for the other protease, the PL-Pro, also some molecules that could do that job, but I would now like to focus on this one because this was in particularly potent and interesting. So what we now could do is we could use our probe to quantify how well an inhibitor actually binds to the active site of the coronavirus protease. And you can imagine that if the probe can bind completely, then you get a very strong fluorescent band. And if you titrate in more and more of this potential inhibitor, then it would fade out. And when you only have a lot of inhibitor, then probably you would see no fluorescent signal at all. And so you can get such a curve and then determine actually how active such a compound is. And that's what we did here. So here you can see that one of our compounds was extremely potent. So here at all of these concentrations, which are quite going to a low range of concentrations, it would block actually the activity of the coronavirus protease or block it from being labeled by our probes. And that's what we also could show here by these concentration-dependent curves where we could see that our best compound, that's this compound A here, is very potent in inhibiting the labeling. And so this is a quite good candidate now, which could be now further used and which also demonstrates the use of our probes as tools to discover novel inhibitors that later on could eventually result in drugs against coronavirus. I don't claim that this compound is now a perfect drug that could be used, although actually it's quite interesting, and it also is already approved in China, but not against coronavirus, but for the treatment of cardiac problems. So actually it would have also quite a beneficial effect on some of the side effects the coronavirus has on many people that are infected. So many die from cardiac arrest or from some problems with the cardiovascular system. And so from this perspective, this drug looks very interesting and will be used then. Of course, we now want to study it if it also has this activity in live cells, and that's what we will do now together with collaborators to figure out if this compound also can block the replication of the virus in human cells. And if that would be still positive as a result, then it would be a very interesting candidate as a future drug against coronavirus. But after all, we have shown that our probes provide a very interesting and unique possibility in finding novel inhibitors against coronavirus, and could be used now in future also to screen more extensive compound libraries and find possibly novel compounds against coronavirus. So to sum it up, the coronavirus requires the activity of two important proteases that are required in order that they can replicate inside the human host. We've developed a novel strategy that allows us to find small molecule chemical probes that are specific to any protease that we want them to be specific for by just creating diversity of probes with a very simple strategy. And we discovered with this strategy some chemical probes that were specific for the active sites of the two coronavirus proteases. We have shown that the proteases are labeled by the probes in the background of human proteomes, which make them useful as chemical tools to further investigate the proteases and what they do in cells and how their activity performs and so on. And furthermore, we have shown that our probes are important tools to find new inhibitors that block the activity of these proteases and could thus be helpful in future for drug development. We thereby also identified one potential candidate that's already approved for human use, and that we will now go further on in testing if this has also activity in live cells. With this, I would like to thank, of course, all of my co-workers, especially those here who did all of the work. So the main part of this project was done by Lilian Penalva in my lab, who, despite of the pandemia, still was always in the lab and did most of these experiments. Then Philip Schmidt and David Samoshvari, who also contributed greatly to this work, and our collaboration partners of the Peters Group, Christoph Globisch and Kevin Savade, and Stefan Schilknecht, who made to us available some proteomes of human cells. With this, I would like also to thank you, and I'm happy to answer your questions.